Welcome to this TMDL presentation for the Upper Chariton River Watershed. My name is Jim Hallmark, and I'm an environmental engineer with the Iowa Department of Natural Resources. And I will be presenting the findings of our Water Quality Improvement Plan for the Upper Chariton River Watershed. This presentation has been prepared and is being presented as part of the public review process for the Upper Chariton River Watershed TMDL. We are currently in a public notice period that will go through August 21st of 2023. If you have any questions or comments regarding this presentation or the Water Quality Improvement Plan, we must receive those comments by the end of the public review period, which, as mentioned, ends on August 21st of 2023. You can send those comments directly to me at james.hallmark@dnr.iowa.gov or to our general TMDL comment email, which is tmdlcomment@dnr.iowa.gov. Again, those comments must be received by August 21st of 2023. Also, if you would like a copy of the Water Quality Improvement Plan, a link to the document will be provided below. Just a couple of housekeeping items related to terminology. The first one is impairment. We use that quite frequently in this presentation as well as in the document. Impairment is defined as a weakened state or weakened condition. It's in a diminished function. So when we're talking about a water body being impaired, there's something preventing it from meeting its designated function or expectations as outlined in the state's water quality standards. In the case of the streams or rivers in the Upper Chariton River watershed, the streams are impaired for E. coli, which is preventing them from meeting their designated function or expectation for recreational use. The other two terms are Water Quality Improvement Plan and TMDL, or Total Maximum Daily Load. These are two separate terms and have different meanings. However, sometimes they are used interchangeably. What is a Water Quality Improvement Plan? In short, Water Quality Improvement Plan is a document, a report, that is made up of a TMDL and an implementation plan. So the Water Quality Improvement Plan is a document, while the TMDL, Total Maximum Daily Load, is an actual number or value. Again, so what is a TMDL? As defined by the EPA, a TMDL, or Total Maximum Daily Load, is the maximum amount of pollutant that a body of water can assimilate while still meeting water quality standards, or while not being impaired. In other words, it meets its intended function and has no diminished capacity. And again, a TMDL is a value. It's a number, it's a target that we're trying to achieve. Here is an illustration of what a TMDL is. Here is a picture of a system, a mule harnessed to a cart. Under normal circumstances, the cart would be loaded with boxes or products, and the mule would pull that around the village or to its destinations, and it would function as expected. However, as shown in this picture, there comes a point in which the load placed on the cart results in an imbalance in the system, causing the mule to be lifted off the ground. In this instance, the system is impaired. It no longer functions as expected. This is certainly a case of diminished function, but all is not lost. The load on the cart can be offloaded or redistributed, resulting in the mule's feet resting on the ground again and the system being restored to function as expected. We can compare this mule cart system to our lake or aquatic systems. The mule and the cart represent our aquatic system, and the load or boxes on the cart represent a pollutant load. Just as with the mule cart system, our aquatic system can handle a certain amount of load or boxes or pollutant and still function as expected. However, there comes a point when the pollutant load reaches a tipping point, resulting in the aquatic system not functioning as expected. There is too much pollution or load on the system, resulting in diminished function. Again, all is not lost. The pollutant load to the aquatic system can be reduced, allowing it to function as expected. So here, what I'm going to discuss for the remainder of the presentation is an outline of our water quality improvement plan. And what we discuss in the water quality improvement plan is a description or a brief description of the Upper Chariton River watershed. We'll talk about how we go about monitoring and assessing a water body. We'll talk about a TMDL development, which includes looking at potential sources of the impairment or E. coli, how we calculate the TMDLs, a brief discussion on an implementation plan that we encourage local involvement. We discuss some best management practices and also continued monitoring of the water body. And also we include a public meeting, and this presentation is part of that public meeting or public review and comment, where we put the document out for public review and comment and ask for those comments from the public. So the Upper Chariton River is in south central Iowa, as can be seen here in this insert map. It's along the Iowa Missouri border. It includes portions of 7 Iowa counties and extends just a teeny bit into Missouri. It has a drainage area of approximately 525,000 acres, or approximately 821 square miles. It has an average annual precipitation of approximately 40 inches. The watershed is divided up into 3 sub-watersheds or 3 sub-basins. There's Wolf Creek, which makes up the northern part of the watershed, the South Fork Chariton River, which essentially makes up the central part of the watershed, and then Cooper Creek, which is the southern end of the watershed just below Rathbun Dam. Each of these subbasins range in size from 237 square miles to 316 square miles. The water quality improvement plan is divided into sections or chapters, with each sub-watershed being its own section or chapter. Land uses in the watershed consist of 8 different classifications or categories. The predominant land uses in the watershed are row crop, grassland, and forest, which makes up approximately 90% of all the land uses within the watershed. And as you can see here, the forest typically is going to follow along the shoreline of streams and rivers. Here's the land use in a pie chart. And looking here at the grassland, we see that the grassland has 281 square miles, and it makes up 34.2% of the land within the watershed. And then we have forest and row crop again. That makes up approximately 90% of the land use within the watershed. Other land uses would include our developed land, which would be cities, roads, farmsteads, anything developed. And then we have water, alfalfa, and just a very small amount of barren land. An assessment of the Upper Chariton River watershed is what's wrong with the Upper Chariton River Watershed. Well, first of all, the streams are not supporting its designated use of primary contact recreational use due to high levels of the indicator bacteria E. coli. So the streams are impaired for E. coli. E. coli limits are established within the Iowa Administrative Code. There's two criteria. One is a geometric mean of 126 organisms per 100 milliliters and a single sample a maximum of 235 organisms per 100 milliliters. And what is E. coli? Well, first of all, it's a large and diverse group of bacteria found in human and animal waste, and it's not necessarily harmful to humans, but it can indicate the presence of harmful pathogens and bacteria and viruses. So why do we use E. coli? Well, first of all, there's just too many pathogens to monitor on an individual basis. So we monitor for pathogens where we have available tests or detection methods that are readily available. And we know that E. coli comes from known sources. If we it comes from the animal and human waste. If we detect high levels of E. coli in a water body, then there is a potential that harmful pathogens are present also. Again, there are just too many pathogens to monitor individually. As a result of this, we use E. coli as a fecal indicator bacteria or an FIB. So how do we go about assessing a water body? First of all, it starts with data collection. We're going to collect water samples and send them to a lab for analysis. Samples are collected over a period of time and are generally collected at the same location. The locations that samples were collected are shown here on this map in orange triangles. You can see them scattered throughout the watershed. We also have identified here red triangles. Those are USGS gauge stations where we gather and collect stream data, flow data that we also used in the analysis of the TMDL. And just for some general information, the weather station here just on the Rathbun Dam that we use just for general information. Samples are collected during the recreational season between March 15th and November 15th, with most collected between Memorial Day and Labor Day. Typically for streams, samples are collected from a bridge or close to one where there is easy access. Samples are collected and sent to a lab to determine the chemical and physical properties of the water body. We're looking for phosphorus, nitrogen, temperature, dissolved oxygen, E. coli, turbidity, and etc. So based on the sampling and analysis performed, there are 16 impaired water bodies or stream segments that are impaired for E. coli, as shown in this figure. A breakdown of these impaired streams shows that there are 8 in Wolf Creek subbasin. There are 6 in the South Fork subbasin, and then there are 2 here in the Cooper Creek subbasin or subwatersheds. So what are some of our potential sources? Potential sources are divided into 2 categories. There's the non-point sources and the point sources and the non-point sources. Point sources are those sources that require a discharge permit, such as an NPDES permit or National Pollutant Discharge Elimination System. The best example of a point source would be a wastewater treatment facility. owned and operated by a municipality where they receive wastewater, treat it, and then discharge it to a receiving stream. These facilities would have that permit that would limit the amount of pollutant that they can discharge. Here is a map showing the point sources within this watershed. We have a general permit number 4, the pink boxes. These are small privately owned treatment systems that have a capacity not to exceed 16 people. So they are very small treatment systems. We have these green triangles, which represent our NPDES permitted facilities, wastewater treatment facilities, and you'll notice that they are close to our cities or communities. Then we have the unsewered communities, the yellow circle with an X through. These are communities in the watershed that do not have a central sewage collection system and treatment system. And technically, unsewered communities are non-point sources. However, for the purposes of this TMDL, they are considered a point source. We've considered them as a point source to allow for future development. So if they ever do have a treatment system, a sewage collection system, that we've accounted for them here. The last one is this burgundy or dark red circle, which are animal feeding operations. These would be facilities that have 1,000 animal units or more, or they have a discharge. They have their permits allow them to discharge to a receiving water body. However, we have none of those within this watershed. So all of these facilities, these animal feeding operations, are non-discharging facilities. And just to kind of summarize all of that, we see we have 13 wastewater treatment facilities, or those that are permitted with an NPDES permit. We have 8 unsewered communities, 10 CAFOs, concentrated animal feeding operations, and 10 of these general permit number 4s, and for a total of 41 point sources within the watershed. The other sources are non-point sources, and there's a variety of those. These would be the sources that do not require a permit, and the best example of that certainly would be a runoff from, whether it be an urban runoff situation or a rural runoff, land application of manure and runoff. So some of the other potential sources within this non-point source would be a direct deposition of manure in streams, grazing animals, wildlife, domestic pets, and failing septic tanks would be some other potential non-point sources that could contribute to the impairments. And now that we've collected data and identified the impaired waters, we can determine the total maximum daily load that the water body can assimilate. Where we want to achieve compliance with water quality standards, and also achieve compliance where the water bodies support their designated uses. And I said in this case, it's recreational use. The TMDL is composed of several components. We've talked about a point source, but we'll also refer to them as a waste load allocation. Our non-point sources be referred to a load allocation, and then we're going to set aside some of our load or our target as a margin of safety. Typically we use 10%, and this is to account for uncertainties within the system. So we can put all that into an equation. Here we have our TMDL, or you might refer to it as a loading capacity, allowable load, and it's made up of the summation of all of our waste load allocations, or the point sources, summation of our load allocations, or the non-point sources, plus our margin of safety. Okay, so since the process to determine our TMDL is the same for all 16 impaired segments, we're going to kind of walk through one. Here we have a stream. This is Walker Branch. It's in that South Fork. And we'll walk through one. Now, this is a very short segment, very short stream, but the process is the same regardless of whether it's short or whether it's really long like this one. The process is the same. So we'll walk through one. We're going to use what's known as a load duration curve, which is a plot of along the y-axis of E. coli in total mass organisms per day, and the y-axis is a logarithmic scale, which means that the difference between each of these units is as a factor of 10. So 1 times 10 to the 7th is greater than 1 times 10 to the 6th, which is greater than 1 times 10 to the 5th. So as you go up, you're increasing by a factor of 10. Along the x-axis is our flow rates, or we'll call it a flow duration, which is a percent. We'll plot it in a percent exceeding or percent exceedance. And I guess what that would mean is that if we have a full set or data set of flow rates, and we pick one and it turns out that it is say a 50% exceedance, then that would mean that 50% of the flows are greater than that individual data point, and 50% are less than that individual data point. And if we move up here to the 10%, we pick a flow rate, and it's associated with the 10% exceedance, then 10% of the flow rates are greater than that individual data points, and 90% are less. And if we come down here to the opposite end, and we have that a data point that's a 90% exceedance, it means that 90% of all the flow rates are greater than that individual data point, and 10% are less than that individual data point. We've also taken this graph and we've broken it up into 5 flow regimes or flow groups, high flows down to our low flows. And that will help us later on determine where our potential sources might be coming from. So we're going to plot our load duration curve. We're going to use, in this example, our 126, our geometric mean of 126 organisms per 100 milliliters. If we were to use the 235, it would essentially be the same process. So here we are. We're going to plot the 126 organisms per 100 milliliters times our flow rate times some conversion factor to come up with this curve here. Now, for each flow regime, we want to establish a target, a goal, and we're going to select for that target goal. We're going to select the midpoint within each flow regime. And that's what our target's going to be within each of these groups or flow regimes. And next we can plot all of our data points. So we see here we've got our summer data points are these brown, and which would be basically from a June 1st to August 31st. And we have a green, which is our spring. See a couple there. That would be from March 15th to May 31st. And then we have the darker color, the black, which would be our fall, which is September 1st to November 15th. So we can plot all of these on our curve. Now we just, for this point here, let's say we have taken a sample. We know what the concentration is on that, for that sample. We look at the average daily flow rate on the day that was is associated with that sample that we collected, and we can plot it at that point. And it just turns out that that particular point is right at about our 50% exceedance. So we know that 50% of our data points are above that point and 50% are below. The same thing up here with this particular one. It's on the 10% exceedance. So we know that 10% of our data points are greater than that and 90% are less than that. So the next thing we're going to do is plot our geometric mean within each flow regime. So looking at all the data points within this wet condition, we will plot the geometric mean, and it will be represented by this green dotted line. And again, that's all the data points within the wet condition flow regime. And we can do the same for each flow regime in our high flows, mid-range, dry, and low flows. And coming back to our wet conditions, we can see the difference between our green dotted line, which is our target, our TMDL, and our green dotted line, which is our geometric mean. And the difference between those two represents the reduction in E. coli needed to achieve water compliance with water quality standards. And we can see here our green dotted line is above the dashed line in our high flow condition or wet condition in our mid-range. But down here in dry conditions, we see that our geometric mean is either coincidental or slightly below our target value. And in our low flow condition, our geometric mean is well below our target value. So these two flow regimes are in compliance with the water quality standards. And then next we can plot our waste load allocation. Remember, these are our point sources, those sources that require a permit to discharge. And those, that discharge of E. coli is constant throughout. Because that's what they're permitted. They can't discharge more than than what's allowed in their permit. And we can see that the difference between our waste load allocation and our target value, that difference will be allocated for our load allocations or the non-point sources and in our margin of safety. So putting all of this in a summary table, here's our wet conditions. Here's the geometric mean, and here's our target value, our loading capacity, our TMDL. And the difference between those two is this departure value. And then that again represents the reduction in E. coli needed to achieve water compliance with water quality standards. And that's an 82% reduction. Just a note here that we're going to select the highest percent reduction as our target area. And the reason we selected this wet value and highlighted it as opposed to the high flow regime is the highs and low conditions are not normal. They are extremes. So we'll look at these three, the middle three flow regimes as normal or typical of values. And so that's why we'll focus on those. And then we talked about our equation a little bit. So the sum, here's our waste loads. It's constant across the board, doesn't change. Then our load allocation and our margin of safety, which again is 10% of our capacity or TMDL target value. And the summation of these three will equal this value. Our last point here, this 6.2 CFS or cubic feet per second, is the midpoint flow rate in that in the wet flow regime, and we have the midpoint flow rates shown in each flow regime. And then just putting that back into an equation form, here's our target or our TMDL value, our waste load allocation or the point source. Load allocation is our non-point source, and our margin of safety. And the summation of all these three will equal to that. Again, just kind of putting it into a different format. Maybe some people like to see it graphical. We have our waste load allocations to here at the blue line and represented in this purple bar. Then we have our load allocations here, and it's difficult to see, but we have right up here the orange. You can see a little bit of the orange line, which is our margin of safety. It's difficult to see because this is a logarithmic scale, and so it kind of distorts things a little bit. But all of the information is here. This gray bar represents the reduction in E. coli needed, excuse me, to achieve compliance with water quality standards. This figure is kind of gives us a general idea of where we would expect our point sources to come from in relation to high to low flow conditions. So as a rule of thumb, if we're looking up here in our high flow, wetter conditions, we typically, as a rule of thumb, are going to look at those sources that are associated with non-point sources. So we have, manure applied to crops and runoff associated with that, as well as manure runoff from grazeland. Grazeland. And then a sanitary sewer overflow, where our sanitary sewers might be old and just don't have the capacity to handle the sanitary sewer and any groundwater infiltration that comes as a result of wet conditions. Failing septic systems here, kind of in the mid-range of things. And then down here in our low flow conditions, we can typically look at our point sources or something that is a direct discharge to receiving water body. That could be, as a direct deposition by animals in streams, from a wastewater treatment plant, an illegal discharge. Somebody's putting something down the storm drain that's illegal, that they shouldn't be putting down. And so again, here we can see on the low flow end, we typically, as a rule of thumb, want to look at the point sources, and up here on the high end side of flows, we typically want to look at our non-point sources. Okay, so then we talk a little bit about our implementation plan, and we provide some a general plan or outline to improve water quality. It includes possible solutions, which would be our BMPs or best management practices. We want to encourage local participation and also allow for future monitoring to evaluate the success of our improvements in the watershed, as well as continue to evaluate compliance with water quality standards. So our best management practices, or BMPs, there's two types of controls. There's a source control. This would usually be something that's a non-structural, where we're trying to limit the pollutant source at its source. Don't want it to expand beyond the point that it's generated at. And then the next one would be an in-drainage best management practice, or BMP, and it can be something structural, but it's treating the pollutant source somewhere downstream from its original point of origin. Few examples of our control strategies: manure management plans, whether it making sure that manure is stored properly, and then when it is applied, that it's applied at the appropriate time, and methods are used to apply it. Pasture management, eliminating stream access by livestock. Septic systems, making sure that our septic systems are in good operating condition, and repair and replace them as needed and necessary. So that's just a few examples of this control strategies. The next would be our in-drainage strategies. Constructed wetlands would be one. It results in settling of particles. It also allows longer time exposure to UV exposure, which can kill E. coli, and then predation, where there's microbes that would break down and decompose the E. coli. And then our vegetative and riparian buffer strips, again, acts as a settling, reducing velocities of runoff, allowing large particles and such to settle out so that they don't make it into our waterways. Okay, so our next steps in this would be to encourage locally led efforts. And this could include the creation of watershed groups where none exist at this time. These groups can promote awareness, and also with local involvement and buy-in, there's always a greater chance for success. With the groups would come along with develop a watershed management plan, and then implement water quality improvements, such as some of the BMPs that I discussed. And then after improvements are made, again, we continue to monitor to evaluate compliance with water quality standards, and to evaluate the effect and efficiencies of those water quality management plans. And then where needed and necessary or appropriate, DNR to provide technical assistance. With that, thank you for watching this presentation. As a reminder, we are in a 30-day public comment period, which will end on Monday, August 21st of 2023. If you have any comments related to this presentation or to the Water Quality Improvement Plan that you would like to make, we must receive those comments by Monday, August 21st of 2023. You can send those comments directly to me at my email, james.hallmark@dnr.iowa.gov, or to our general TMDL comment email, which is tmdlcomment@dnr.iowa.gov. And again, those comments must be received by Monday, August 21st of 2023. Again, thank you for watching.